who doesn't know Tamil, please raise your hand. You will find out now. There may be some Tamil words and which we can explain in English. Otherwise, no, the presentation is going to be in English only. There are requests from audience that they want to have it in Tamil. Okay, only a couple of people.
Two participants raised hand.
Yeah, we'll cover everyone.
Okay. We will definitely adjust for that.
I think we should start, no?
Yeah.
I think we should start.
Yeah, it's 5 minutes over. Okay. Friends, so after a gap of 3 months, so we are starting the PSL again. This time it's going to be continuous because we stopped because of the second wave of coronavirus. Of course, none of us are none of the organizers are not getting affected, but no, we know that the audience and their family members might be affected. So in case of organizing such meetings, the family members may be sick and they cannot attend the meeting. They feel they may be missing it. So that's the reason we want to put it on hold for some time till we get back to at least normal life. So now we feel that it is the right time to start it. And today we have a lecture on post-COVID-19, COVID-19 created or evolved. There was a discussion in the media as well that the COVID-19 has been created in the lab. Some scientists has also been proposed some theory to support this kind of argument. So we are going to deal with such arguments today, and so there are two lectures. One is going to be delivered by Dr. T.R.G. He will deal with about how does the theory will withstand the scientific scrutiny. The other one will be delivered by Dr. S. Krishnasamy. He will go a little bit detail into that and biology of the coronavirus and how whether it can be made in lab or not, something like that. He will deal with that. So with that, we have Dr. Ramanujam here to organize the meeting. He's going to be the moderator. In the meantime, I request you to fill up the feedback form. So we are very much interested to get your feedback, and not only the feedback, we want to get your contact details, particularly the mail ID or email or the mobile number, so that we can communicate with you about our future programs. So we don't have any mechanism to collect it in the online meetings. Only thing is the feedback form. Please fill up the feedback form. If you don't have any feedback, at least you fill up your details, your name and the email ID. That's enough for us to communicate with you in future. So with that, let me hand over the mic to Dr. Ramanujam.
Thank you, Vijay. Since he introduced the topic in English, I'll follow up in Tamil so that we have this balance. நண்பர்களே அனைவருக்கும் வணக்கம். தமிழ்நாடு அறிவியல் இயக்கத்துடைய பொது அறிவியல் உரைகள் வரிசையில் இன்றைக்கு ஒரு முக்கியமான ஒரு கருத்து எடுத்துக்கொள்ளப்படுகிறது. அதாவது என்னன்னா, நம்முடைய இந்த கொரோனா பெருந்தொற்று நம்ம நம்முடைய வாழ்க்கையில ஒரு மிகப்பெரிய பாதிப்பை ஏற்படுத்தி இருக்கிறது. அந்த வைரஸ் எங்கிருந்து தோன்றியது, அதோடைய தோற்றம் எவ்வாறு என்ற கேள்வி கிட்டத்தட்ட நம்ம எல்லார் மத்தியிலையும் உண்டு. இதை ஜூனோசிஸ்னு சொல்லுவாங்க. இந்த வைரஸ்ங்கிறது விலங்குகளிலிருந்து மனிதர்களுக்கு வருவது என்பது. அதை பொறுத்த வரைக்கும் ஆடியோ கேக்குதா? அப்ப அவங்க ஏதோ பரிமளா நீங்க செக் பண்ணிக்கோங்க. அப்போ இந்த வைரஸுடைய தோற்றம் பற்றி பல கேள்விகள் எழும்பி இருக்கின்றன. இன்றைக்கு ஊடகங்கள்ல இது பற்றிய பல விவாதங்கள் ஏற்பட்டு கொண்டிருக்கின்றன. பலர் சொல்வது இது இந்த வைரஸ் என்பது இயற்கையிலேயே தோன்றியதா அல்லது சைனாவில் இருக்கக்கூடிய வைரஸ்க்காக இருக்கக்கூடிய ஒரு ஆய்வகம் ஒன்று, அந்த ஆய்வகத்தில் தோன்றியதா, அது அவங்க வேணும்னே வெளியே விட்டாங்களா இல்ல விபத்தால் வெளியே வந்ததா என்றெல்லாம் பல இந்த மாதிரி கேள்விகள் கேட்பது ரொம்ப சாதாரணம், தேவையும் கூட. ஏன்னா உலகத்தையே பாதிக்கக்கூடிய ஒன்று நடக்கும் பொழுது கேள்விகள் எழுப்புவது மிக முக்கியமானது. அறிவியல் இயக்கத்தை அறிவியலை பொறுத்த வரைக்கும் கேள்விகள் எழுப்புவதன் மூலம்தான் நாம் உண்மைகளை தெரிந்து கொள்ள முடியும். இந்த கேள்விகள் கேட்பது மிக அவசியமானது. அதே நேரத்துல அந்த கேள்விகளுக்கு வழி தேட வேண்டிய ஒரு வழிமுறை இருக்கு பாருங்க, ஒரு அறிவியல் வழிமுறை, அது ரொம்ப முக்கியமானது. ஏன்னா இந்த கேள்விகள் பின்னால பலவிதமான அரசியல் பின்னணி இருப்பது நமக்கு தெரியும். என்றைக்குமே அறிவியல் அன்றாட வாழ்க்கையை தொடும்போது அரசியல் கலந்து வருவது ரொம்ப இயல்பானது. அறிவியலுக்கும் சமூகத்தில் இருக்கக்கூடிய தொடர்பு என்பது எப்பவும் இருக்கக்கூடியது. இதுலேயும் பலவிதமான சமூக அரசியல் பின்னணிகள் இருக்கின்றன. இந்த அடிப்படையில இருக்கும் பொழுது இப்போ விஜயன் சொல்லும் பொழுது கூட சொன்னார், இதற்கான விடைகள் மட்டுமல்ல, அந்த விடைகள் நமக்கு எவ்வாறு தெரிகின்றன. அதாவது இந்த நிறைய தியரி சொல்லலாம், இது இங்கேன்னு வந்தது அங்கேன்னு வந்தது இப்படி வந்தது. ஆனா எந்த தியரியாக இருந்தாலும் இறுதியில அதற்கான அறிவியல் விளக்கம் மிகவும் தேவை. சிலவற்றிற்கு நமக்கு ஒண்ணு தெரிஞ்சுக்கணும், அறிவியல்ல எல்லாவற்றிற்கும் இன்றைக்கு விடை சொல்ல முடியலன்னா அது அறிவியலுக்கு புறம்பான விஷயம்னு கிடையாது. இன்றைக்கு என்னால விடை சொல்ல முடியவில்லை என்று அறிவியல் பூர்வமாக சொல்வதும் அறிவியல் தான். நமக்கு இருக்கக்கூடிய சாட்சிகளை வைத்துக்கொண்டு, ஆதாரத்தை வைத்துக்கொண்டு இன்று நமக்கு என்ன தெரியும் என்று விவாதிப்பது. அதற்கு இன்றைக்கு நமக்கு வந்திருக்கக்கூடிய இரு சிறந்த விஞ்ஞானிகள், பேராசிரியர் கோவிந்தராஜன் அவர்களும் பேராசிரியர் கிருஷ்ணசாமி அவர்களும் இது பற்றிய அருமையான ஃப்ரண்ட்லைன்ல ஒரு அழகான ஒரு கட்டுரை எழுதியிருக்கிறாங்க. அந்த கட்டுரையை நீங்க வாசிக்கணும்னு கேட்டுக்கிறேன். இது பற்றிய விவாதம் நடத்தவும் இங்க வரக்கூடிய பல கேள்விகளுக்கு விடை விவாதிக்கவும் வந்திருக்காங்க. இதே நேரத்தில் நாம் ஒரு குறிப்பிட்ட இந்தியன் சயின்டிஸ்ட் ரெஸ்பான்ஸ் டு COVID-19 என்ற குழுவை பற்றி உங்கள் மத்தியில் சொல்ல விரும்புகிறேன். இது கொரோனா பெருந்தொற்றுக்கான இந்திய விஞ்ஞானிகளின் கூட்டமைப்பு என்று கடந்த மார்ச் மாதத்திலிருந்து பணி செய்து கொண்டிருக்கிறது. இது பற்றி, இந்த குறிப்பிட்ட கேள்வி பற்றி ஆழமான பல விவாதங்கள் சில மாதங்களாக நடந்தன. அதுவும் நம்முடைய நண்பர்களுடைய இதுல இருக்குன்னு நினைக்கிறேன். ஸோ தட்ஸ் ஆல்சோ அ கான்ட்ரிபியூஷன். அது ரொம்ப முக்கியமான கான்ட்ரிபியூஷன். ஏன்னா இந்த மாதிரியான விவாதங்கள் நடப்பதன் மூலம்தான் நமக்கு தெளிவு ஏற்படுகின்றன. அப்படிப்பட்ட விவாதங்கள் மிக அவசியம். இது அறிவியல் இயக்கம் விவாத மேடைகளை அமைப்பதில் பெருமை கொள்கிறது. அறிவியல் ரீதியான கேள்விகளுக்கு விவாத மேடைகள் அமைப்பது நமக்கு மிக அவசியம். அந்த அடிப்படையில் இந்த PSL 36 ரொம்ப ஒரு முக்கியமான ஒரு பணி செய்கிறது என்பதை நான் மிகவும் பெருமையுடன் சொல்லிக் கொள்கிறேன். ஏற்கனவே சொன்னது போல மொழி நமக்கு ஒரு இடையூறாக இருக்க வேண்டியதில்லை. whoever is comfortable with English, we can discuss in English. whoever is comfortable in தமிழை தான் ஆர்கியூ பண்ணணும்னு சொன்னால் தமிழில் பேசலாம், அது ஒன்றும் பிரச்சனை இல்லை. நம்மளால இரண்டுத்தையும் மேனேஜ் பண்ணுறதுல என்ன பிரச்சனையும் கிடையாது. அனைவருக்கும் நன்றி. மீண்டும் அனைவரையும் வரவேற்று பேராசிரியர் கிருஷ்ணசாமி அவர்களை விவாதத்தை தொடங்குமாறு கேட்டுக்கொள்கிறேன். ஸோ ப்ளீஸ் எஸ்கே, ப்ளீஸ் ஸ்டார்ட் ஆஃப் த டிஸ்கஷன்.
ஓகே. நண்பர்களே வணக்கம். என்னோட ஸ்கிரீன் டைட்டில் ஸ்கிரீன் தெரியுதுங்களா? நான் ஷேர் பண்ணியிருக்கேன். சரி. இப்போ நான் இது அதான் நான் அவரு தமிழ்லயும் இங்கிலீஷ்லயும் சேர்ந்து பேசினா ஓகே தானே? அல்லது
SK, that may not be very effective. சேர்ந்து பேசுறது ரெண்டு பேருக்கும் புரியாம போலாம்.
Couple of people don't know Tamil. அத பாத்துக்கங்க.
Okay. So though the title says I will be talking on the biology behind coronavirus, நான் அதை பத்தி பேச மாட்டேன். I will be talking more about virus in general and how whether the COVID-2 virus could have been made artificially or leaked in the lab, or could it have more likely evolved. So I will place the arguments for that, and Govindaraj will continue, TRG will continue with that in a more broader perspective. So I'll just go to my next slide. That is on, can you see my next slide?
Yeah.
அடுத்த slide தெரியுதுங்களா?
Yes.
Yeah, okay. So it's 11:15. So I will try to be done by 11:25 or by 11:30 to 40. So if I'm exceeding that, please just indicate on that. So what I have tried to tell here is that there are a lot of viruses. No, if you look at it, in the 10 nonillion viruses now, that comes to 10 power 31. So that is 1 followed by 31 zeros. So another analogy is to say that for every star in the universe, if you can assign about 10 crore viruses, then that will be the total number of viruses on Earth here. But obviously they all don't infect humans. If they were all infecting humans, we will not be there. Though our being here is because of viruses. It is because of virus insertion only that mammals allowed fetuses to be present in the body and then get rejected. So that is a mammalian protein. Origin is a virus. But viruses from bacteria, plants, they don't infect humans. That is why when you eat plants, even if they have viruses, we don't get infected. Whereas if you look at mammals and birds, they have about 17 lakh viruses which are not yet discovered in them. That's the estimated one. So there are a huge number of viruses which can infect us. So we are still going to get more viruses infecting us and pandemics, and obviously there's no need to create. There is enough naturally present for people to get infected. Now, what is known is there are about 200 viruses that causes disease in humans, but not all viruses which cause disease in humans started off that way. They generally viruses are present and they are parasites. They need a host organism, a cell, and once they develop in that cell, then they can go to other type of species or cross barriers. If they develop the required proteins and mechanism needed. So obviously one virus attaches to a cell because of a receptor. It's like a handshake. So rather than use a lock and key analogy, which is very rigid, it's more like a handshake that a virus, protein on the virus shakes hand with the protein on the cell surface, and thereby allowing the virus to enter the cell. So if the cell happens to be amongst these bats or rats or chimpanzees or gorilla, they will infect them. And you can see most of these viruses, the cases which you get are from these animals which are shown on the left side here. On the left is the number of cases you get. And you can see how the virus progresses. You can see this is an estimated curve showing how a virus, what is shown at the bottom pink color is the virus infects, say, a gorilla and then goes off after some time and again infects other gorillas and so on and keeps going. So it cycles within the bat or within the rat or within the chimpanzee species like this. However, at times when there is an intersection between, say, bats and the domestic animals, or rats and domestic animals, then the virus can spill over, meaning it can cross-infect into the domestic animals. For example, if a bat dropping is eaten by a pig, or a bat bites a cow, it could be because of it has to be some liquid material which gets transmitted across. So the virus can get across. It can be even by respiratory, as the current COVID-2 virus or the SARS virus. So once it gets into the domestic part, the large green one, we have a huge amount of domesticated animals. The largest number of animals which we have on the earth is from domesticated animals. So they can get into the domestic animals, and you can see that gets amplified and goes on. So, for example, it may get infected into a pig, and then it may get into a bird, or it may get into a cow, and so on. So there can be transmission between them also. Again, for that, they have to develop the appropriate proteins or things which are, after all, we all have a common ancestor. No, so we are all linked together. So the protein in us, hemoglobin protein in us, is also there in other mammals, is also there in other species are there. So there are a lot of commonalities. So it's just a question of choosing the right receptor so that they can go into other species. So that is called a spillover. Now, if his spillover happens in domestic, it's not too much of a problem, except, for example, when there was a rinderpest virus, entire cattle got infected, or even now recently the pigs that were killed, or the chicken that were killed because of the influenza virus affecting them. So at times, because of the domestic animals interacting with humans, the spillover can happen either from the domestic animals to humans, or it can happen from the virus in the host which is carrying. Chimpanzee, for example, can come into this. So then it gets amplified within the humans because of interaction between the humans. Sometimes viruses need vectors, like, for example, mosquito or a parasite, or a tick, like that. It can get transmitted. So sometimes, even though the spillover happens from one human to another human, or from one domestic animal to another, a vector has to be involved to transmit the virus. But in the case of respiratory viruses like common cold and SARS and so on, we don't need such vectors. Or also for HIV, blood or saliva, the transmission between body fluids can do that. So you can see, and it's a periodic thing. It's not that it goes away. It keeps happening all the time. So every 2 weeks or so, you have the virus cycling inside these animals, and then there's a larger cycle within the domestic and a smaller within the human. So that's why we get periodically, about 2-3 months or so, there will be some virus infection that happens, and then it goes away and comes back again, so on. So this is a continuous process. What I have shown on the right side here are the viruses which we know: smallpox, measles. Smallpox and measles are ancient diseases. Now it's been shown by reconstruction using DNA that they are from ancient mummies also, in the BC things. But then, even though we make so much of hue and ha about the SARS-CoV-2, we still do not know what is the actual intermediate host of smallpox or measles. We know measles probably came from some ancient rat, and smallpox from some ancient cattle thing. But we really do not know what is the actual intermediate host which it occupied before coming out to humans. And same thing with the influenza pandemic. Influenza pandemic of 1918, which happened earlier, which is referred to now. There also, even though the influenza virus that actually caused the pandemic has been reconstructed in parts and in whole by people, they still do not know the full origin. It's thought to be coming from some bird, but which birds? What is the actual intermediate host? That is still not identified. And if you take Marburg virus, this is the most deadly virus that causes hemorrhagic fever. 1967, the Marburg city in Germany, there was a sudden incidence of this hemorrhagic fever and people died. And then they thought that, okay, it's coming from some monkeys which are imported for research. Maybe that monkey, people are working with it, they contracted it. And now it has been traced to bats. No, bats coming in Uganda from Africa. So the hypothesis is that these bats somehow transmitted to some monkeys, and those monkeys, when they came there, they transmitted the virus to this thing. But even now, which bats and how they are getting transmitted, there's a population of bats that's supposed to be in a cave in Uganda, which people are actually tagging to see how they spread, how the bats spread and how they may be trying to find out. Occasionally, the Marburg virus incidents happens, people die, but there is no clear thing of how the virus is getting. Is it only directly from bats, or is there an intermediate host? Only for the case of HIV-1, the 1981 was the first incidence of HIV AIDS disease in New York. So immediately people didn't start saying, okay, New York, some conspiracy, people infected people in New York. There's some Russians infected them or Chinese infected them in New York and something. No, for 10 years, people didn't know really, that they thought maybe some chimpanzee was there. No, finally, but still the chimpanzee virus did not have enough relationship. Like what they're talking about the SARS now, even much larger variation was there. Finally, a chimpanzee, a small chimpanzee, which was sent from Africa to US, shipped when it was a kid. And then after 20 years, when people were screening the chimpanzees in captivity with the research, among 98 chimpanzees, one of them, this Marlin, that chimpanzee kid named Marlin showed the antibodies to HIV-1. But Marlin had not been infected at all with HIV or had got transmission, blood transmitted. So finally, they figured out that there is a chimpanzee, pan-triglyceride PTT, which has a simian chimpanzee virus, which is the closest relative, and therefore that must be the intermediate host. But it was not causing HIV in the region where this chimpanzee was living in Africa. No, much later, because it got saying it. So this time between finding the actual intermediate host with definitive sequence evidence and making the connection can take a long time, like in the case of HIV, 20 years. But people didn't start saying that HIV-1 as a conspiracy, it was produced and infected by Russians into US and so on, even though the first occurrence was in US. No, and same thing with SARS. Even though the SARS virus, which first showed up in China in 2002, people initially thought they are from civet cats and palm cats, because those were shown to have viruses which are similar. And then people who are handling these palm civets, they were found to be also having infection. But now more recent evidence in 2015 indicates that probably SARS directly came from bats, because a huge amount of bats which are there, which are having relatives which are very similar to the SARS virus. So that civet population or palm civet population might have helped only in the amplification of getting, because they found that the initial people who were infected by the SARS, they were not getting as much, because SARS uses also the same ACE2 receptor as COVID-2. But after passaging through the palm civet, somehow the virus seemed to be better at infecting the humans. So because of passage through the civets, it seemed to have become better at infecting humans. But that was not the actual place from which it jumped to the humans. So, right now the immediate idea is that SARS also originated from bats, and it has been passaging in bats, and then it has jumped directly from bats, likely. MERS, Middle East respiratory disease, this happened again. No, the first in Middle East, obviously that's where the name came. And within a year of that, people, within about 9 months, people said, okay, one guy who was having this MERS virus, saying the camel which he was handling, they found that camel also had an identical MERS virus. And so they jumped to the thing that, okay, this is because camels, dromedaries and camels, that is how the MERS gets transmitted. But now people have shown that from camels, you cannot really very easily infect humans from thing. So camels don't seem to be the real host from which it has jumped to the humans. Maybe it got infected because of, just like nowadays the tigers and no, in Vandalur Zoo also the lion or tiger got infected, or in New York Zoo, same way maybe, or how the minks got infected in Finland and Denmark, same way this camel probably got infected. But it seems to be unlikely that that is an intermediate host. So now for SARS-CoV-2, it's hardly a year. And now people are jumping, saying, okay, because SARS-CoV-2, we don't have a real intermediate host, definitive host, therefore it must be artificially created in humans. So you can see this whole thing is resting on very flimsy evidence, and all likelihood, just like in the earlier pandemics, this is probably a natural evolution of the virus, which has allowed it to jump from either bats directly into humans, or maybe through some other domesticated animals into humans, but most likely bats into humans. Okay, so now if I go to the next slide, pardon? There's a question or somebody asked something?
No, you can proceed with it.
No.
Okay, so
just SK, there is a question on chat.
I can't see the chat.
Okay, no. Without infection, how are antibodies found in monkeys?
Yeah, because there's a cross reaction. That is the simian chimpanzee virus is so similar to the HIV-1 that antibodies raised in the actually I didn't tell that part completely. That is, what happened was, just soon after they did the serosurveillance of the chimpanzees, that marlin died giving stillbirth to twins. No, and so probably it was infected by SIV and it developed into this thing. And this SIV PTT antibodies to that cross-react with the with the HIV thing. So that is this thing. So the antibodies are not really antibodies which are made against HIV-1, but because they are made against the simian chimpanzee virus PTT, they are so similar, they cross-react with the thing. Just as, for example, human coronavirus, earlier things cross-react with the current SCoV-2 antibodies virus also. Yeah, so coming back to my slide here, this is just to show that, you know, what people have shown now is that the zoonotic viruses, that is viruses which cross over from animals, they are mostly present in rodents and bats. You can see the large number of present in rodents and bats. The Lassa virus is a classic example coming from rodents. It's a very dangerous virus, Lassa virus. But again, it's thought to be from rodents. I mean, it's shown to be from rodents, but it doesn't seem to directly transmit from humans to humans. So that way it's safe. It's still not got into a pandemic. And what is shown on the right and top is showing that if you look at the abundance amongst the domesticated animals and amongst humans, humans are everywhere. No, and therefore, obviously, they have become very good sources for the virus to infect. So from rodents or bats, it's very likely that they jump to humans, because more and more humans infringe into ecosystems and occupy a lot of ecosystems and niche areas. So that is why humans are a very good potential host for viruses, and therefore you can expect more and more pandemics and viral pandemics because of the sheer fact that humans are infringing in all eco spaces. So this is an estimate of this paper in 2017, where they have estimated after report bias correction, showing that the most likely places for origin of zoonotic diseases, including viruses and bacteria, are China and the Indian subcontinent. You can see here. No, that's because obviously here there are a huge amount of people, and the forests are much less. So the propensity of other animals which are there for getting transmitted are much less. So emergence of new diseases are most likely from China and India. And for example, what happened recently in China for the SARS, and again for Nipah and Kerala, probably because China has a much better surveillance system, they were able to detect the cases much faster than you can imagine what would happen if such things happen in Bihar or UP, for example. it would have gone undetected for a large amount of time. So this is just a nice chart showing how the pandemics have happened over the period of time. And you can see that most of them are bacterial diseases, bubonic plague and so on. They're controlled because of antibiotics now. However, smallpox and Spanish flu, they are from viral things. And as I said, amongst all these other things, the one which has a very clear intermediate host is HIV, and that was the AIDS, HIV-1, and that was identified only after 20 years. So, Ebola, for example, we still do not know what is the actual thing, and it's thought to be bats as a reservoir, but what exactly is happening from there, we don't know. Swine flu, the same Asian H1N01 it's called PDM09. That's because there's a small bit of the swine, the porcine or pig virus, which seemed to have been found in the 90s, that has got inserted into that. But why is there? Is it in that, and how did it? Did it actually come from pigs, or what happened? We really do not know about that. So COVID deaths are shown here. It is actually initially here when the poster was prepared in April 20, but now the more number we have is around here. So hopefully it will not go in this case here. This is just to show you that this intermediate host part is still not very clear in this. And if you look at this is from GSA, where there are about more than 2 million sequences which are sequenced from various people who have been infected and deposited here. And what primarily to show you that we know all these variants which are very commonly seen now. The reason I've shown you this is that saying that the SARS-CoV-2 was a perfectly designed for human infection is not really true, because obviously it has started changing from day one onwards. You can see this huge amount of variation that has happened, and what is shown at the bottom is all the sequence changes that have happened over the entire genome. This is the entire genome, about 30 kilobases. You can see almost every part of the genome there have been changes in nucleotides. This is a spike protein region, and where more changes are there. But obviously, if it is so engineered and perfect, why should it show so much of variation, and that too right from the early stages? So obviously, it's not a thing. There are enough cases now, and we'll come back to this furin business later. There again, you will see there are so many changes happening that in some of these cases, the furin doesn't actually cleave. There are viruses which are immune to that, which are resistant to furin cleavage. They obviously exist because they have been sequenced from human samples. All right, so just coming back to this is a phylogeny tree from the same sequence next strain where you can see, just to show you that if you look at this SARS-CoV, and these are bat viruses from bats related to SARS-CoV. This SARS-CoV-2 at the bottom red here from various human beings, and what is the closest one from bat here, and also the rat G13 that was reported in the Nature paper initially, right? So you can see that what is important is the horizontal distance. The vertical is just for separation. So you can see the horizontal distance between this rat G13 and the human ones, or the what was seen in the pangolin and the humans, is much smaller than what you see between the SARS-CoV and the bats reservoir that is there. So if people can accept the SARS-CoV came from bats, why can't they accept SARS-CoV-2 came from from other bats or other reservoirs? So this is, in fact, shows that we found hosts which are having much closer sequence relationship to SARS-CoV-2 than SARS-CoV, right? So this COVID-2 definitely is of bat origin. And how it came, whether it circulated in bats and then jumped directly into humans, or it was circulating in humans in a milder way and then finally became more prevalent and it was detected, that is not clear. But obviously it's a thing. So now coming to the last part, the furin. This is that furin is a protease that is there in all. So you can see that it's used in a lot of places in the human body, in mammalians. It's used by many viruses. It's used by many bacteria as also for this thing. So the furin protease, herpes, other coronaviruses, even HIV has two domains, and the furin cutting that loop only enables the entry into the human cells. So there are many such viruses which get activated by proteolysis and specifically by furin. And so to say that a furin protease site was engineered into the SARS-CoV-2 begs the question, I mean, would it really be the case when you have so much of convergent evolution happening? And that is what Darwin said way back, when he talked to the eye, where there are so many instances of convergent evolution of the eye, that he said, look, just because the eye seems so well designed and so well adapted, and you cannot say that it is goes against the theory of evolution. It is just a creation, because he said there are so many intermediates which you may not be able to capture, right? So same thing here. So this is almost my last but one slide, just to show you that this is from the paper that is shown here, zenodo.org. It's a preprint which is just published in July, which these figures are from that. Just before I come to the figures, I'll just show you on the left-hand side what's shown here. There are many proteases that help SARS-CoV to enter the cells, right? So the major one is called the TMPRSS2, transmembrane serine proteinase 2, right? And of course, elastase, trypsin, and furin. You can see what is shown at the bottom here, how in the absence of getting cut by TMPRSS2 or any trypsin, which will allow the merging of the membranes to happen, there are other ways of getting in. There's an endosome formation and can get in. So there are alternate pathways. So this is one pathway in the absence of any protease cleavage through the endosome formation. Another pathway is by cleavage of trypsin or with TMPRSS2. Usually, furin occurs inside the Golgi bodies inside the cell. And furin cleavage of the COVID-2 spike protein helps in the COVID-2 spreading from one human cell to another human cell more easily, right? Rather than the first entry point. That's why I told you earlier there are many sequences where the furin cleavage site is lost, and yet they are infecting because it uses TMPRSS2 to infect. But spreading from one human to another human cell is also aided by furin. So absence of furin in no way makes COVID-2 of any that it cannot cause a disease. So obviously engineering furin site is a very silly thing to do. No, the most obvious likely thing is that it's evolved naturally, because you can see in many beta coronaviruses, what is shown here in red, this PRR, right? This is the what is called by this Nicholas Wade, as Govindarajan will say, as a smoking gun, because of what virologist in Baltimore said, and Baltimore subsequently withdrew that comment. No, so this PRR is the one which is there. You can see that many of these viruses have that. The important thing is the arginine at the bottom, the right side here. This is just shown for reference, the green one C as a reference point. You can see these are all insertions which have happened in various things. The MERS is there. So it's like saying MERS-CoV has a furin cleavage site. SARS-CoV doesn't have a furin cleavage site, but SARS-CoV has a ACE2 receptor binding. So it's like saying a green guy wearing a green shirt comes and robs a house, and then a guy wearing a red pant comes and robs the house, and then you get a third robber who is wearing a green shirt and a red pant, and therefore immediately you say, okay, this green shirt and red pant are new things. It's not new things, right? They're already there. So if you look at the top, what is shown here is the thing that what these people have shown and others have also shown is that the virus is not this insertion that has happened. If you take with respect to the bat virus, RATG13, that is a reference virus which was used in the initial Nature paper, where the report, see again, China reported the sequence of the virus within 2 weeks of it being determined and identified. Why should they share the sequence if it's a conspiracy? No, because they shared the sequence of the virus, subsequent PCR, RT-PCR and vaccine development became much easier. They could have said, okay, you go and find out if it's going to be a conspiracy theory. So here, what is shown here is on the top part is that the insertion is not an insertion as would happen if you were to cut and paste. It's not like that. It's actually a small, you can see the serine codon here, that one. It's called -1, -2. So as you may all know, codons are written threes, 1, 2, 3, 1, 2, 3, 1, 2, 3. Right. So you can either cut after 1, 2, 3 and insert a set of codons which will code for a set of peptides like this RRA, PRRA, and put it in. Or what happens naturally is that it doesn't cut after 3, but either there's a cut after 2 or a cut after 1. So it's actually it changes the reading frame. So instead of 1, 2, 3, 1, 2, 3, it goes 1, 2, 3, 1, and then something else comes in, and then the 2, 3 happens elsewhere here again, right? Or it can come after 2. So such sort of a slippage or insertion happens only in recombinations, not with engineered. Now, this is a very complicated way of engineering, which nobody would be doing such things. So this is all which is there. I will just tell the last part about the lab leak. The lab leak, now once people kept coming with these arguments, immediate thing was saying that, okay, it was not created artificially, but then no, the Wuhan lab is such a poorly kept lab that there was a lot of leakage from there. No, lab leaks can happen anywhere and so on. What is interesting is actually lab leaks have occurred earlier, and all were for viruses which were identified to be infecting humans, and then they were experimented in the lab, and then they leaked from such things happen in Russia, in China, and US. In fact, there was a earlier thing to sign a biological treaty to look into such things to prevent such accidents. And guess who didn't sign that? Who said no, we don't want to sign that? USA, because USA said no, we don't want to sign all such treaties, right? And now they are the ones who are now accusing that China has made this leak on this from Wuhan. And what you can show, what is seen here is on the topmost panel are the sequences that happened immediately after the thing was shown. You can see a lot of divergence which are seen in here. No? And what is shown here is the Wuhan Institute of Virology here. This is a Yangtze River. And these are the Hunan market from which initial identification or the other markets from which identification. This is a distance of about 20 kilometers. Like saying that Guindy King's Institute is doing some research, and then the infection happens somewhere in Kasimade near Royapuram, right? So Kasimade Fish Market, there is a thing. So this distance is something similar here. So you don't see such thing reporting at all here, no? And if you look at the infection cases that have been identified, they're all far away from what the Wuhan Institute is here. And if you look at even the infections that were reported, the excess deaths due to pneumonia, which maybe are unidentified infections, you can see the initial in January, the deaths are reported way on the other side of the Yangtze River, not this side here. No. So Daniela Anderson is a person from Australia who has worked in Wuhan. She's the only foreign scientist who's been working there in Wuhan. And what she has gone on record is to say that the records in Wuhan are clean. No, they were tested. Nobody got infected with the SARS-CoV-2 or something similar. During that period, she was there till about around this time in November also. And she said there's nothing strange. And the level of safety is far higher than what other labs she has seen elsewhere. No, so if at all there's a lab leak, actually it should go to the US, because the first infectious clones where they had COVID COVID engineered to have a furin site was done in Cornell University, and that was about 4-5 years back. And also in Chapel Hill University, which was collaborating with Wuhan, they made the infectious clone there of engineered SARS-CoV having a furin site, and that was then tested out and passaged in Wuhan. No, so obviously if the leak could have happened, it should have happened either from Cornell or from Chapel Hill, USA. But nobody is talking about that. So If you're talking of lab leaks, it could have happened anywhere. It's very unlikely to have happened in Wuhan, looking at all the cases that have happened. But then if there's a fair and broad inquiry, let the inquiry cover all of them. Let it cover the US, let it cover other places. But as I said earlier, US didn't want to sign a treaty which allowed people to look into their labs. So how are they expecting other countries to start doing that now? So obviously there's more motive to it than a simple thing. And at least in terms of biology, all sources indicate that the virus is naturally evolved. Intermediate hosts maybe can be found only after a huge screening of the bat population is done across various places. And the virus is not perfect because it's been showing a lot of variations. And the furin site existed in other viruses, coronaviruses also. And the rare codon that is alleged, the codons are rare, but they do occur in beta coronaviruses. The CGC codon occurs about 3% of the time in beta coronaviruses. So it's not rare. And obviously, if it was some engineered, and it should have got mutated, but that is an area which has not got mutated at all. There's about 99% or 98% in all the 2 million sequences that those codons are conserved in across the thing. So obviously, it's a very stable entity, and it's very unlikely to have got engineered into this part. So that with that, I'm completing my part.
SK,
yeah.
நீங்க கன்க்ளூட் பண்றதுக்கு முன்னால இங்க ரெண்டு கேள்விகள் இருக்கு. ஒன்னு வந்து is furin used in the vaccines as a component? அது ஒரு கேள்வி இருக்கு.
Yes, what?
Furin, Furin, furin.
Okay. Yeah.
Furin used in vaccines. இன்னொரு கேள்வி என்னன்னா, are antibodies formed because of actual COVID virus and by different vaccines the same antibodies formed by
so
COVID virus
furin,
as well as vaccines?
So the first question, furin is a protease. It's a molecular scissor that cuts other proteins. This protease is not present in vaccines, or is it? It's present only in the inside human cells and in other mammalian cells. No, so furin is not there in any of the therapeutics that are used, including vaccines. The other one, that if you take antibodies produced by vaccines, like the ones against the spike protein, for example, the Covishield or the mRNA vaccines, more of them are produced against the spike protein, right? Whereas antibodies that happen against infection during an infection tend to happen against all other proteins which are exposed by the virus. So, for example, the antibodies produced against in Covaxin vaccine and other inactivated viral vaccines are more likely to be similar to the repertoire or the set of antibodies that are produced against the proteins that are exposed by the natural viruses. The vaccines which use only the spike protein to raise antibodies obviously make antibodies only against the spike protein.
SK, one more thing. The last two slides la நீங்க சம்மரைஸ் பண்ணி விளக்கினீங்க இல்லையா? அதாவது இது லேப் இது இல்ல, இந்த விளக்கத்தை மட்டும் தமிழ்ல கொஞ்சம் சுருக்கி சொல்ல முடியுமா? ஏன்னா
ஏதாவது நான் என்னன்னா வந்து அந்த கடைசியில காட்டினது வந்து ஏன் லேப்ல லீக் ஆகாதுங்கிறதுக்கு ரெண்டு இருக்கு. ஒண்ணு வந்து அங்க இந்த அது நான் சொல்லல, அது ஒரு பேப்பர்ல போட்டிருக்காங்க, ஜூலைல வெளியிட்ட ஒரு பிரீப் வந்திருக்கு. அதுல என்ன, அது பல ஆத்தர்ஸ் வெவ்வேறு நாட்டுல இருந்து அதுல இருக்காங்க. அதுல என்ன சொல்றாங்கன்னா, அந்த வூஹான் இன்ஸ்டிடியூட் ஆஃப் வைராலஜி இருக்கிற இடத்தையும் அந்த ஹூனான் மார்க்கெட் அங்க வந்து மொதல் கேசஸ் ஐடென்டிஃபை பண்ணது பார்த்தா அது கிட்டத்தட்ட ஒரு 20 கிலோமீட்டர் தூரத்துல இருக்கு. அங்க அந்த வந்த கேசஸ் எங்குமே வூஹான் இன்ஸ்டிடியூட் பக்கத்துல ஐடென்டிஃபை ஆகல. அதே போல வந்து அன்னோன் நிமோனியான சில பேர் எக்ஸஸா சில பேர் செத்து போறாங்கன்னா அவங்க அந்த மாதிரி நடந்தது கூட அது யாங்ஸி ரிவருக்கு அந்த பக்கம் இருக்கிற அந்த வூஹான் மார்க்கெட் அங்கதான் வந்து இருந்திருக்கே தவிர இனிஷியல் டேஸ்ல இங்க வரல. அப்போ வந்து இங்க வூஹான் இன்ஸ்டிடியூட்ல இருந்து வெளியில ஒரு என்ன இந்த இதுவா அங்க வேலை செய்யறவங்க எதான கொட்டிட்டு அல்லது இதுல இருந்து லீக் ஆகி வந்ததுன்னா பக்கத்துல இருக்கிறவங்க தான் நிறைய இன்ஃபெக்ட் ஆயிருக்கணும் அப்படி ஆகல. அதே போல வந்து அவங்க அந்த டேனியல் ஆண்டர்சன் அவங்க அங்க வேலை செஞ்சிருக்காங்க ஊஹான்ல, ஆஸ்திரேலியால இருந்து ஒன்லி ஃபாரின் சயின்டிஸ்ட் அங்க வேலை செஞ்சவங்க. அவங்க என்ன சொல்றாங்கன்னா, ஊஹான்ல இருக்கிறவங்க எல்லாரையும் அப்ப செக் பண்ணாங்க, யாருக்கும் ஆன்டிபாடிஸ் அகேன்ஸ்ட் SARS-CoV-2 இல்ல, அப்போ இன்ஃபெக்ஷன் ஆகலன்னு அர்த்தம் அவங்களுக்கு. அதே போல அங்க இருக்கிற சேஃப்டி ஸ்டாண்டர்ட்ஸ் ஆர் வெரி ஹை அண்ட் ரெக்கார்ட்ஸ் எல்லாம் கிளீனா இருக்கு, அதனால சஸ்பெக்ட் பண்ண வேண்டிய அவசியம் எதுவும் இல்லை அப்படிங்கிறாங்க. அதுல நான் கூடுதலா ஒரு தகவல் என்ன சொன்னேன்னா, இங்க வந்து நீங்க இந்த SARS-CoV-2 வந்து அந்த ஃபியூரின் கிளீவேஜ் சைட் இருக்கு, அதுதான் வந்து அதுக்கு வந்து குற்றவாளின்னு சொல்றாங்க. அது ஆக்சுவலா இது இல்லை, ஏன்னா அது SARS-CoV-2 ஃபியூரின் யூஸ் பண்ணுதுன்னா மீதி வைரஸஸும் அதை யூஸ் பண்ணுது, மெர்ஸும் யூஸ் பண்ணுது, பல பீட்டா கொரோனா வைரஸும் யூஸ் பண்ணுது. நான் காட்டின மாதிரி பல நம்ம உடம்புல இருக்கிற பல இதுல ப்ராசஸஸும் யூஸ் ஆகுது, பாக்டீரியா ஆந்த்ராக்ஸ் மாதிரி கூட யூஸ் பண்ணுது. அதனால அது ஃபியூரினை வந்து யூஸ் பண்றதுங்கிறதுல இது அர்த்தமே இல்லை. இதுல அந்த கோடான்ஸுங்கிறது வந்து சிஜிசிங்கிற கோடான் வந்து 2 வாட்டி வந்திருக்கு, அது வந்து அங்க யூஸ்வலா ஆகிறது இல்லைன்னு சொல்றாங்க. ஆனா அது அந்த சிஜிசிங்கிறது வந்து பீட்டா கொரோனா வைரஸஸ்லயே நிறைய வாட்டி இருக்கு. அப்போ கிட்டத்தட்ட 3% இருக்கு. அப்படி இருக்கும்போது அதுல நேச்சுரலா வர்றதுல இதுல இருக்கு. அந்த அப்படி வெளியில இருந்து கொண்டு வந்து போட்டிருந்தா யூஸ்வலா வந்து இப்போ கட் பண்ணி போடும்போது நம்ம கோடான்ஸ் எல்லாம் 1 2 3 1 2 3 1 2 3 படிக்கிறாங்க, 3 3 படிக்கிறாங்க. அப்போ கட் பண்ணும்போது என்ன, 1 2 3க்கு அப்புறம் கட் பண்ணி புதுசா ஒரு ஏதான இன்சர்ட் பண்ணிட்டு மூடுறது தான் இன்ஜினியரிங் இதுல பண்ணுவாங்க யூஸ்வலா. ஆனா இதுல எப்படி இருக்குன்னா, 1க்கு அப்புறம் ஒரு வெட்டுப்பட்டு அந்த 2 3க்கு நடுவுல ஒரு புகுத்தல் இருக்கு, அல்லது 1 2க்கு அப்புறம் வெட்டுப்பட்டு அதுக்கப்புறம் புகுத்தல் ஆன மாதிரி இருக்கு. இது மாதிரி நகர்றது வந்து ரீகாம்பினேஷன் ஒரு ப்ராசஸ் இருக்கு. டிஎன்ஏ நம்ம இதுல வந்து ஒன்று சேர்ந்து பிரியறது. அந்த மாதிரி நடக்கும்போது அந்த மாதிரி யூஸ்வலா நடக்கும் அதுலன்னு. அதனால இது மாதிரி இப்படி இன்ஜினியர் பண்ணும்போது இவ்வளவு காம்ப்ளிகேட்டடா பண்றதுக்கு ஒரு வாய்ப்பே இல்லை. அது மட்டும் இல்லாம இது வந்து வெளியிலேருந்து புகுத்தப்பட்டதுன்னா, இப்போ இந்த 2 மில்லியன் இது சீக்வன்ஸ் பண்ணதுல வந்து மாறுதல்கள் நடந்திருக்கணும் அந்த இடத்துல. ஆனா மாறுதலே நடக்கல. அந்த சிஜிசிங்கிறது வந்து 98% அந்த 2 மில்லியன் சீக்வன்ஸ்லயும் இருக்கு. அப்போ அது வந்து ஒரு ஸ்டேபிளான ஒரு இன்சர்ஷன் நேச்சுரலா நடந்ததுன்னா நம்ம பார்க்க வேண்டியது இருக்கு.
ஹலோ சார். பட் இதே நம்ம பார்த்தோம்னா அதே பேப்பர்ல ஒருத்தர் ரிப்போர்ட் பண்ணிருக்காங்க, இந்த ப்ராஜெக்ட் எடுத்ததே வந்து NIH தான் அப்ரூவ் பண்ணிருக்கு இந்த ப்ராஜெக்ட்னு சொல்லிட்டு நம்ம படிச்சிருக்கோம் சார் அதே பேப்பர்ல. ஸோ தே வாண்ட் டு பர்டிகுலர்லி இன்சர்ட் த ஃபியூரின் கிளீவேஜ். ஆமா இல்லையா சார்? அதே பேப்பர்ல எழுதிருக்காங்க.
இல்ல அதாவது அவங்க கெயின் ஆஃப் ஃபங்க்ஷன் ரிசர்ச்ங்கிறத வந்து வந்து அலோவ் பண்ணாங்க யூஎஸ்ல. அதுக்கு பிறகு தடை விதிச்சாங்க. அதுக்கப்புறம் அதை வந்து இது சொன்னவொடனே மறுபடியும் அலோவ் பண்ணாங்க. அந்த கெயின் ஆஃப் ஃபங்க்ஷன்ங்கிறது பொதுப்படையா ஒரு வைரஸை வந்து எப்படி மேம்படுத்த முடியும் அப்படின்னு ஃபியூரின் பண்றதுன்னு இல்லை. நான் நடுவில் சொன்ன மாதிரி இந்த ஃபியூரின் அந்த கிளீவேஜ் சைட் ஆக்சுவல் ஒரு ப்ராப்பர் ஃபியூரின் கிளீவேஜ் சைட்டை அந்த SARS-CoV அதோட சீக்வன்ஸ்ல இன்சர்ட் பண்ணது கார்னல் யூனிவர்சிட்டியில பண்ணாங்க. அது வந்து கிட்டத்தட்ட 5-6 வருஷம் முன்னாடி பண்ணாங்க. அவங்க வந்து அது எப்படி வந்து அந்த புரோட்டியோலிசிஸ் வேலை செய்யுதுன்னு பார்க்கறதுக்காக பண்ணாங்க. அதே போல வந்து சாப்பல் ஹில் அவங்க தான் வந்து அந்த பாரிக் அப்படிங்கிற அவங்க கொலாபரேட்டர்னு அந்த வூஹான் இன்ஸ்டிடியூட்டோட இருந்தாங்க. அவங்க தான் அங்க ஃபர்ஸ்ட் SARS-CoV-ய இன்ஃபெக்ஷியஸ் குளோனா ப்ரொடியூஸ் பண்ணி COVID-2 மாதிரி அங்க ஒரு குளோன் ப்ரொடியூஸ் பண்ணி அங்க அந்த குளோன்ஸ் தான் பிறகு இங்க கொண்டு வந்தாங்க. அப்போ லேப் லீக் நடந்திருக்கணும்னா கார்னல் யூனிவர்சிட்டிலேயே நடந்த வாய்ப்பு இருக்கணும், அல்லது நார்த் ஹில் அங்க கேரலினால அங்கேயும் நார்த் கேரலினா சாப்பல் ஹில்லையும் நடந்திருக்க வாய்ப்பு இருக்கணும். ஏன் அப்ப நீங்க லேப் லீக்னு பொதுப்படியா பார்க்கும்போது ஏன் அந்த இடத்துல சேர்க்கல? அப்ப யூஎஸ் லேப்ஸும் இன்வெஸ்டிகேட் பண்ணணும் இல்லையா? ஆனா யூஎஸ்லயே முந்தி வந்து இந்த மாதிரி லேப் இன்வெஸ்டிகேஷன்ஸ் அந்த ஒரு பயாலஜிக்கல் சேஃப்டி இதுக்கு வந்து நாங்க அந்த ட்ரீட்டிக்கு கையெழுத்து போட மாட்டோம்னு சொல்லியிருக்காங்க. அப்போ அது எப்படி கரெக்டா இருக்கும்? நீங்க வந்து இங்க சைனால வந்து நடந்திருக்கும், அங்க வந்து லீக் இருக்குன்னு. அதுக்கு இந்த டேனியல் ஆண்டர்சன் அவங்க வேலை செஞ்சிருக்கோன்னு சொல்லியிருக்காங்க. நான் அங்க இருந்தேன், இந்த 2019 நவம்பர்ல அங்க இருந்தேன். அங்க வந்து நல்ல ரெக்கார்ட்ஸ் தான் வச்சிருக்காங்க. அங்க யாரும் வந்து எதையும் மறைக்கல. அங்க வந்து எல்லாமே கிளீனா தான் இருக்கு. அங்க யாருக்கும் கோவிட் ஆன்டிபாடிஸ் டெஸ்ட் பண்ண போது இல்ல அப்படின்னு. அப்போ இதெல்லாம் பார்க்கும்போது இதுல வந்து நமக்கு ஆதாரம் இல்லைன்னு தான் தோணுது.
தேங்க்ஸ் எஸ்கே. நான் என்ன நினைச்சேன்னா, பேசும்பொழுது சில விஷயங்கள் அப்பப்போ தெளிவு வேணும்னா நம்ம நடுவில் எடுத்துக்கலாம். விவாதங்களை இறுதியில் வச்சுக்கிறது சரியா இருக்கும். ஏன்னா பேராசிரியர் கோவிந்தராஜனும் பல கருத்துக்களை சொல்ல போறாரு. அதுலேயும் இந்த கருத்துக்கள் திரும்ப திரும்ப வரப்போகுதுன்னு நினைக்கிறேன். அதனால தேங்க்யூ எஸ்கே. அடுத்து பேராசிரியர்
சாரி, நான் கொஞ்சம் நேரம் ஜாஸ்தி நேரம் எடுத்துட்டேன்னு நினைக்கிறேன். ஓகே.
ஸோ லெட்ஸ் அடுத்து பேராசிரியர் கோவிந்தராஜன் அவர்கள் இந்த தன்னுடைய கருத்துக்களுக்காக சொன்னார்.
என்னுடைய ஸ்கிரீன் தெரியுதா?
தெரியுது.
Okay. So I'm going to speak in English. My write-up will be in Tamil, and I will try to mix both of them in my way of presenting. In fact, SK has done a great job in bringing the background of the virus itself and pandemic, and at the end he also went into some questions about the controversy between lab created or evolved. And let me give the final summary, at least to me, and probably since I have talked to SK and we have written an article together, the final summary is it is very, very, very likely evolved. And if it is lab leaked, it is a serious question which international organizations should start playing, how to record various events in labs and getting without ignoring any country, because it is a serious question, because whatever happens in one country will happen in other countries. Similar things, for example, on issues of nuclear question, etc., there are some agreements, and people have to agree to certain events of this kind, that they will be subjected to, all the countries will be subjected to the issues about how the virus, etc., spread, because it is not related to one country, and it is like something which is going all over the world, and already it is still many people have died, and several crores of people have been infected, and we are hopefully, by the developments of vaccine and also social, the better understanding right now, unlike the last year, we may be able to overcome by probably middle of next year to a satisfactory level. Till then, many of these problems will continue to be there. So this is what I was mentioning. This is about Wuhan Virology Institute, and the question is Wuhan. I have written Mahanam, it's not correct thing. Wuhan city, which is in Hubei. The question is, who is the first patient? And the declared one is 2019, December 30th, from Wuhan seafood market. And they found some rare event, or the way in which this pneumonia was attacking that person. And the reference was given to the Wuhan Institute itself to analyze this thing. And they did a great job. And within short time, they gave the sequence for the World Health Organization. And at that time, it was not even declared as a pandemic. It was limited to Wuhan alone. And then we asked the question, where did it come from? How did it come there? And the answer is still to some extent uncertain. We don't know, but we have a good understanding as of now, much better than what was there in 2020 January. Now, after this, when the world woke up, because earlier they thought it is a problem of China and it is a problem of Wuhan, And the world woke up after South Korea developed several places, in a place in south part of South Korea, coming from a church event. And again, later in Italy, where certain area in the northern part of Italy was affected. This was in early January and early February. In fact, late January and early February, even though 1 or 2 cases were reported in other places. Now, in Italy, this thing actually went into the question. A lot of people, particularly senior people, started dying. In fact, they were so much affected that they had to make a choice. Who will get the benefit of the ICU? Because they have to make a choice. That kind of situation developed. Later, Spain, and then other European countries also. Now, one thing is very interesting. When it happened, because many of us had friends in Italy and Germany and other places, we came to know about their problems. And in February 2020, actually February 28, popular science lecture in our place, which is the same thing which we are having. Popular science lecture was the first lecture organized on this issue of SARS-CoV-2 virus for the first time in the whole of India. In fact, I have never come across this discussion about how this virus, and the first meeting, public meeting, we held in Anna Library. In fact, our YouTube link is still there, and we held, and there were 2 people. One was a person who was associated with Salem Government Hospital. He was a person who actually was involved in RT-PCR testing later in March and April in Salem Government Hospital labs. And another lab diagnostic center person, Pavitra, her name is Pavitra. She also took part in that meeting, and we were the first to start that. And very interestingly, we had a student from Wuhan itself attending that meeting, because he came to know about this meeting. He had returned from Wuhan, and of course, a lot of testing was done, and he was found to be RT-PCR negative. And in spite of that, at that time, he was asked to go for quarantine for 14 days, which he did. And then again, another test was done and found to be still negative. So there was no problem. He also attended our meeting, and he conversed with many of us, and we also learned a lot of things, even though the thing is, by last year March, our knowledge of this problem, and I think was very weak all over the world, but we were also late in this thing. We may not realize in the parliament, our deputy, our vice president, in the parliament, in the Rajya Sabha, when some people were wearing masks to come to the Rajya Sabha meeting, he instructed the mask should be removed. So that was the lack of knowledge we all had at that time. And of course, later, a lot of ways in which it was handled, a lot of confusion, whether it should be done that way or not, etc. We held a lot of Zoom meetings after that, but because that was more or less the last meeting we held in public. And after that, it was only Zoom meetings, and at least some 10 meetings, if I remember correctly, between 7 to 8 meetings, at least, we held on this issue. Okay, so this is the famous Wuhan Institute for Virology, and it's a fairly big institute. Definitely about 1,000 people are also working. There are various centers in the neighborhood, and this is an international institute to some extent, because there are people working from US, there are people working from Australia, South Africa, Germany, Switzerland, and there are people who went from India also. There is famous center called National Center for Biological Studies, which is under Department of Atomic Energy, under Tata Institute of Fundamental Research. There are a couple of people who were involved in collaborating with this institute precisely on the nature of the virus, particularly bat, as a source of coronavirus. And they were actually playing a role in analyzing, which I'll probably mention later. So what happened? So after that, various things have happened. And the issue of politics, as Ramanujan mentioned, came into the forefront. And that was the time of Donald Trump was competing for his presidency again. And in fact, he had the support from our Prime Minister also at that time, who went to US and gave his support. And Donald Trump came to India in February, end of February. Remember, this is 2 months after the whole event taking place. He came to India and he even claimed, I have not seen such a crowd in my lifetime. And that was held in Ahmedabad Stadium. So about a few lakhs of people attended. And of course, that was the situation. Now, by about March, April, end of April particularly, by middle of April, the US was affected and it spread, this disease spread to several countries, including US. And what shocked people was this famous event that there are the dead bodies in New York City, in hundreds of people dying, and they were being buried in a mass burial in New York City itself. And the whole of US started reacting and calling Trump as a worst possible leader because he didn't even start taking preventive actions for this thing. That was a political scenario which happened. And naturally at that time, he had to evade his problem and accusations which were happening. So he needed an enemy. And the question was that of virus. Against all norms, in fact, World Health Organization has a norm that we will not blame any country for a pandemic of this kind. It could have originated in US, it could have originated in this thing, like the earlier Spanish pandemic, virus pandemic, which was there 100 years back. So we will not blame any country because then people will start attacking those Chinese, but you call it as a Chinese virus, people will start attacking Chinese who are moving in the street. They have nothing to do with the whole thing. And this is a kind of a situation which can happen. So prevent that, World Health Organization has a norm that we will not name any country. And this is that is why it is called SARS-CoV-2 virus. And Trump accused virus was responsibility of China. And then he called it as Chinese virus, and he even developed this famous theory that HCQ, that hydrochloric, this medicine, and he wanted India, which was developed, which is quite capable of giving this medicine, to thing, and we in some sense benefited to some extent by giving them a lot of this medicine. So then after that, he was defeated in the elections because by end of 2020, 21, this thing, he was defeated. And what happened was Joe Biden came and he started taking actual steps to prevent, and also he insisted masks should be worn and various preventive things should be done. And also we cannot forget, in the current world scenario, China as a very developed, from a very, very poor country in 1950s, has become, by their own management of science and technology and interactions with people inside and outside, a great country for industrial development and also scientific developments. So this is the kind of a thing. So he, instead of saying the Chinese developed this virus like the way Trump was telling, but he also encouraged people to come with the remarks that it came by mistake, by tavarudalaga, by lab leaked. It is not intentional, but lab leaked. This is the kind of a thing. In fact, such a thing was answered develop. And if there are questions, I'm sure me and SK will answer more about it. And this is what happened. All the scientists who were associated with World Health Organization and most of the virologists at that time didn't believe all these statements. They were very clear that the SARS virus, everything came from that bat thing. So there could be certain transfer from bat virus to human beings, maybe through intermediary animal, or as SK was telling, there could be a direct transfer. We do not know. So there are questions like pangolin could have actually played a role in this thing, and we do not know. And the whole question now is, what if it is intermediary? What is that animal? We have to find out, and the scientists will go through that. They will systematically ask the thing. They will do the experiments. They will find the sequence, everything, and try to understand. And it is a very tough task. Let me make it clear, it is a very difficult task, and it will stay as a question for several years, definitely. Now, the genetic makeup of the pangolin seem to be in good agreement with up to 96-97% with the SARS-CoV-2 virus, but still not conclusive. So this is the pangolin. Pangolin is actually, in Tamil, people call it irumbu tinni, and this has a lot of scales. And in China, it is very it is a delicacy, and that's why that Wuhan market, seafood market, where exotic animals are sold, and this is also something which gets sold, and the people felt that it might have played a role in this thing. Now, this coming to this lab, Wuhan Institute for Virology, it is actually to analyze virus. It is a lab which is created to analyze and understand virus, because China was one of the countries which were affected by SARS, and it has been there for some time now. And that lab is actually what is called Biosafety Lab 4. In all these labs, there are categories which are there. The labs are created with some categories. How much safety, double the safety, triple the safety. You want to make sure that any error is not going to affect, in the sense of there are 3 times or 4 times the safety. And that kind of a lab was built by French company in China, in this place, and it's also associated with the university there, and that a lot of students from India also go and study in Wuhan, particularly those who are interested in medical areas. So this is a biosafety lab number 4. This is the highest. This is the highest characterization, number 4. In fact, we do not have, there is only one lab in India which is of this type, which is in Pune. That is where the Pune Virology Institute Virology Lab is the only one, and which finally confirms whether your RT-PCR, whether it is a doubted, finally confirms that this particular thing is correct. For example, National Center for Biological Studies in Bangalore, which is a very famous institute and one of the famous great places for many of the researchers, it does not have a lab of this level, level 4. It has only level 2. That's the comparison just made, because the safety aspects are being questioned, and this we cannot forget. If this kind of a thing is going to happen and lab leak is going to happen, it requires much more stringent procedures to be laid for all the countries, and that's where we have to be careful about it. Now, in this research center, there are various scientists from various countries. As I mentioned earlier, there are people from US, Singapore, Germany, India, Australia, France, Germany. Germany I've written twice. So this is what the kind of people who are working there. And NCBS have some agreement, understanding with doing this work. And in fact, one of the person gave a talk in famous meeting. It was held in IMS in Math Science recently. And this is precisely about the bats and the viruses which are there and how they analyze it. And there are some questions raised which I will not go into that. So to say that Wuhan lab is responsible for this is like scaring people. And I have written in Tamil, they are scaring people by calling Wuhan lab. So we used to do that in since I was involved in neutrino project. People will scare people by saying Fermi lab kind of thing. So I have written explicitly in Tamil, you are showing some puchandi against this thing. We have to ignore it. And this is the way in which this Wuhan Safety Lab 4 people work. Look at the way in which the whole technicians, scientists who have to work, the kind of thing which they have to adopt to do any activity inside the thing. It is completely sensitized, sanitized, everything. So the lab leak is a very, very difficult one. And if it happens, it gets recorded. If any problem happens, any event happens, even if there is a power cut in some small area, which is not in this thing, it gets recorded, but it may not affect the lab because it is permanently supplied by this thing. And similar to whatever happens in nuclear safety labs also. So this is what is a thing. So we would like to know what is the truth. Okay, that is a question. And we have to definitely do more research. As SK was telling, the 2 theories as of now, whether it came from the lab or it evolved, what is evolved seems to be having a much more powerful chance, something like more than 99% or so. But still there is a lack of that 1%. Uncertainty is there. So they have to fix it. That is a part of the research activity at the international level. It is not related only to China. There are about 2 million virus deformations have taken place. And we have to analyze it and try to understand from what happened in that place as well as other places. And this is very important. And an organization like World Health Organization is a coordinating one, and it can play a very important role. So as SK mentioned, all the informations which World Health Organization as a committee asked with China, this lab has been given. There is a possibility some delay could have happened, and I'm not aware of anything which has been asked, which has not been given by this thing. And they tried to pose the China as a villain, and which is not at all required. But here it is a question of lab. There are people working in that, scientists working in that, and they are as much conscious about this thing as anyone else is. So there is a report which has been given by World Health Organization, and it explicitly says that it is coming, it came most likely from evolutionary changes which has been taking from the virus by the virus from bat to human beings, and we do not know that how the intermediate steps have taken place. And they also say very clearly there is no evidence as of now. There is no evidence as of now for the statement that it was changed, the virus was modified to become a pandemic kind of problems, so that the entry into the human cell will be successful. And there is no evidence towards that, or there is no evidence that it leaked from that lab, because there are no events which are recorded there which tell you this. But this is the status. If they come with new remarks that it came from that, then we have to understand it much more carefully. So a lot of it became a journalistic science correspondence thing. They asked several virologists several questions, and depending on what they say and putting their own theory into their mind, they put out various news items. And I just want to give you the background. Gemma Johan, who has actually worked in Wuhan, is a New Zealand virologist. As you know, New Zealand was one of the countries which successfully managed after the virus in that country, very, very successfully. In fact, people used to say several women leaders were handling it much better. They said about Kerala, the famous teacher, Shailaja teacher. And also they said about the president of New Zealand, who was successfully handling, and the president of New Zealand was relying on Chennai, who was a special virologist and advisor for the president, and definitely explaining what happened and how to handle the situation, both as a public health activity as well as a medical activity, and how people should be treated, etc. She was playing a major role. She said that so far five coronaviruses have been there, and they have actually intermediary animals, and there is the spike protein binds effectively in this case, and that the development of that effective binding, the evolutionary distance between the original one and this is so large that it is too difficult to be imagined as made in the lab. This is a famous remark she made, that evolutionary distance is too large. It is very, very difficult to say that this distance can be covered with this distance by manipulating something like that. So the glycoprotein, which helps a virus bind effectively to the receptor ACE2, which was a thing which actually she mentioned that. So how did it help? So there are several people. I'm going to only name 2 or 3 of them. David Baltimore, who was a famous Nobel laureate, I will mention it. And there is an article written by Nicholas Wade, science correspondent, and I will mention where. He also interviewed David Baltimore either through the phone or directly. And he made some remarks, and it was projected by Nicholas Wade in his article, and which was used by Balram and our own scientist from Bangalore, to emeritus scientists from Indian Institute of Science, biologists, who had written a current science article, almost giving some credibility to this claim of Nicholas Wade. So I'm going to mention something about that. The Science as a journal has published a letter immediately after this. And now what is required is a proper scientific investigation and proper scientific thing in understanding this issue. And they also clarified very likely that it is bad to a human being through intermediary or evolved nature of the virus. So next is the Lancet also made very clearly that data sharing is very, very important for this international pandemic which is going on. And if such rumors and disinformation is there, accusing some lab, accusing some country, etc., it will be threatened by this. So people should avoid such remarks, and it should be left to the scientists to come to that. Political interference of this kind for political benefits will not be helpful. Now, the conspiracy theories will quote several places. They will actually quote several places. There will be articles there, New York Times, this, that, etc. But when you all look into that, they will all finally go into one source. And that one source in this case turned out to be an article, well-known writer, science correspondent by name Nicholas Wade. He had written in Bulletin of Atomic Scientists. Now, this person has some dubious record as far as scientists are concerned, particularly biological scientists or genetic scientists are concerned. And he has a book, popular book, which is called Troubling Inheritance, with about genetics and race, etc. And it had a lot of racist overtones. And he had interviewed several people. And what he has done is he had put his own conjectures in issues about race and other intelligence, etc., in his book, which troubled many scientists who gave interviews. And there are about 130 scientists from Stanford who openly wrote a letter to New York Times simply saying that Wade juxtaposes incomplete and inaccurate data and informations to pose conjectures. We reject his book and also his conjectures. The next person they started attacking recently was Dr. Fauci. It is again related to the political difficulties of Donald Trump vis-a-vis Joe Biden. And he was actually this Dr. Fassi was heading the CDC, which controls the virus kind of diseases in US, and he has seen 8 presidents. He was actually CDC for 8 presidents, starting from George Bush onwards, even earlier. So he had seen all these people, and they started attacking him because, as from his department, he gave some funding to an organization, genetic organization called Eco Alliance, which collaborated with Wuhan Institute for Virology. I will mention something about it. So this is the third person who was getting attacked by all these people was Tedros, who was the director of World Health Organization, and it is here. Now, Nicholas Vade, what happened was bulletin in that thing, he actually quoted Baltimore saying precisely the one which actually mentioned by SK, and if there is question, he will also take it up again, that there is this smoking gun, which is that furin, which is there, which could have played some role in this thing. This is a remark which he made. Now, immediately this man took it and made it into a famous theory, and that protease, which was playing the role, and it could have come from the lab. This is the kind of a remark which he wrote. Following that, our Balram also made almost agreeing remarks in current science. But what happened is, after 1 week or 10 days, that famous Baltimore who gave the interview, he realized that his remarks have been misquoted, taken out of context, and he said that Vade has changed my remarks and to fit his convenient conjectures, and I'm actually going away from his conclusions, and I will not make that thing. When I said smoking gun, we have to be careful. This is an important role, but don't think that it has been manufactured. This is the kind of a thing which Baltimore himself made after that, and came out of de-linked himself from that interview. So Fassi, as I mentioned, Dr. Fassi, he was what they call Totrunoi Adhigari, actually CDC, okay? And he allowed Eco Alliance but $6 lakh for doing some collaborative work with the Wuhan Institute for Virology. And because of that, he was rejecting this particular theory that lab was responsible. Now, this is actually a ridiculous thing that such grants have been made. Even Indian government has made grant for research activity, particularly about bats. And that lady who did the work, she did work with Wuhan, her name is Uma from NCBS. She did work in Arunachal Pradesh thing. There's a famous article which was written in Hindu, if I'm right, about the bats which are there in Arunachal. There are caves full of bats, and they were studying it. And she actually went there. The people there, the tribal people there, they smoke, they send smoke inside that cave and get the bats out and catch it and make food out of it and eat. This is the tradition which has been there. And she was studying such people as well as the bats. Samples were taken, the virus samples were taken from there, and their antibodies in that thing. And they wanted to find out whether because the bats are source for several viruses of this type, so they wanted to study. And such grants have been given from India also by actually Department of Atomic Energy. And you can't say that they were doing some work on this kind of a nature. But Fassi clarified. He said he didn't say, even reject the famous theory that it didn't come from the lab. He was said that if you ask me, he actually explicitly said the evolutionary way in which it came to human has a highest chance as of now from whatever data which is available. And he also mentioned there is no evidence so far from that it has leaked or manufactured in that lab. This is the famous remark he made. And he actually had to face some problem. Finally, in Forbes article, the whole thing was summarized by Ethan Segal. He said, people said this is a bioweapon. The coronavirus 2 is a bioweapon. Number 2, Farsi is directly involved in this. Third, that bat woman called Shi Sheng Li was the brain behind all these things. And the coronavirus research is banned in several labs, and that is what they were doing, what is called gain of function work, and this is banned, and that is being done in that lab. There is no evidence for all these things. Now, what has happened is through the virus coming through an intermediary animal or directly from bat seems to be the most reasonable one as of now, which has a very high chance of survival. This is the bat woman who is being accused by these people in that lab, with whom actually Indian scientists and Australian, Singapore and other scientists have worked, and they have said they found nothing specifically incorrect or wrong things being done in that lab. So what is the lesson for us? The lesson for us is this: all the scientists, all the medical people, virologists will agree that this virus is spread all over. It has become a pandemic. It has also led to a collaboration at the international level. The lab information from any place, from India, from US, from Europe, China, all these things are getting distributed, and the collaboration work which is happening voluntarily between all the countries is enormous, and this is one of the things which has happened. And people will realize that Darwin still rules the world of cell biology, and is the thing which people will, as of now, accept among the scientists, and that is the end of the story. So this is what I wanted to tell. I will conclude now. That is this remark, which is the lesson, and we have learned a lot of lessons. We have learned a lot of lessons in the way. It is not like last year, April, where we don't know how to treat. We don't know how to handle. Now the understanding is very good, and people are learning. Medical people are learning, virologists are learning, epidemiologists are learning, and their understanding is far better. But still, the last word is yet to be said on this thing. So I'm going to stop it here, and we will go ahead and further take up questions and other things to both me and SK. Thank you. Thank you, Rajan.
அனைவருக்கும் நன்றி, வணக்கம். I think ஏற்கனவே சாட்ல நிறைய விஷயங்கள் விவாதிக்கப்பட்டு கொண்டிருக்கின்றன. நிறைய பேர் பல கேள்விகளை எழுப்பியிருக்கிறாங்க. பேராசிரியர் கிருஷ்ணசாமி அதற்கு விடைகளும் கண்டு கொண்டிருக்கிறார். வேற இப்போ I think we are almost caught up with all the questions so far. இப்போ புதிய கேள்விகள் இருந்தா அன்மியூட் பண்ணிட்டு டைரக்டா கேட்கலாம்.
ஜாம் அந்த क्वेश्चंस படிச்சிடலாமா? சில பேர் சாட் பார்க்காம இருக்காங்க.
இல்ல சாட் கேள்விகளுக்கு அவர் பதில் அப்பப்போ கொடுத்துருக்காரு. சாட் படிக்கலாம். ஏன்னா ஒரு 30-40 கேள்விகளுக்கு பதில் வந்திருக்கு.
அந்த 2-3 பேருக்கு பதில் கொடுக்காத கேள்விகளுக்கு படிக்கலாமா?
பதில் கொடுக்காத கேள்விகளுக்கு தெரிஞ்சதுன்னா சொல்லுங்க. பார்த்த வரைக்கும் அவர் அப்பப்போ
you can have a look at and tell which are the questions.
அதுக்குள்ள மற்றவங்க கேள்விகள் இருந்தா அன்மியூட் பண்ணி
raise the hand and then ask the question.
raise hand feature இருக்கு. முதல்ல
இருக்கு.
அதனால கை தூக்கிட்டு அப்புறம் பேசலாம்.
சார் ஒரே வாட்டி லாஸ்ட்ல கன்க்ளூட் பண்றீங்களா? What is the conclusion? Is the lab created or evolved?
கேள்வி சரியா கேட்கல, கொஞ்சம் உரக்க சொல்லுங்க.
என்ன கன்க்ளூஷன் கேக்குறாரு? evolved or created?
ஒரு வார்த்தையில சொல்லுங்க.
Conclusion. Okay, Krishnasamy can tell. Actually, the most likely it is from the natural evolution. Very little chance from for the other one. That is the.
Yeah, I would say it is naturally evolved. But if lab leaks have to be curtailed, it has to be done across the world internationally, not only using this as a excuse.
Yes.
So
I would agree with that.
Yeah.
சார் பட் அப்போ இன்டர்மீடியேட்ஸ் எதுவுமே நம்மளுக்கு சொல்ல மாட்டேங்கிறாங்க இல்ல சார். ஃபர்ஸ்ட் நம்ம நோட் பண்ணது வந்து பேட் கொரோனா வைரஸ் தானே சார். அங்க இருந்து திருப்பி SARS-CoV-2க்கு ரெண்டுத்துக்குமே டிஃபரன்ஸ் பார்த்தா நடுவுல இன்டர்மீடியேட்ஸ் நம்ம பார்க்கணும் இல்ல சார். பிகாஸ் ரீகாம்பினேஷன் வந்து சடனா ஒரு க்ளீவேஜ் சைட் வந்து கிரியேட் பண்ற அளவுக்கு அதுக்கு நாலேஜ் இல்லை சார். ஸோ ஒன்னொன்னா ஒரு சின்ன காம்பினேஷன் ஆஃப் அமைனோ ஆசிட்ஸ் ஃபார்ம் பண்ணி ஓகே இது நம்மளுக்கு மேட்ச் ஆகுதா அந்த மாதிரி தானே சார் அது ரீகாம்பினேஷன் கூட நடக்கும். ஒரே வாட்டி ஒரு க்ளீவேஜ் சைட்டை கொண்டு வந்து நடுவுல வச்சுட்டு ஓகே திஸ் இஸ் குட் ஃபார் அஸ், இப்போ இது நம்ம நேச்சுரல் செலக்ஷனை வச்சுக்கலாம், நம்ம நெக்ஸ்ட் கண்டினியூ பண்ணலாம், அந்த மாதிரி ஒரு இது நடக்காது இல்லை சார்.
இல்ல அந்த ரீகாம்பினேஷன்ங்கிறது வந்து நேச்சுரலா நடந்துட்டே இருக்கு. இப்போ ஈவன் வித்தின் ஹியூமன்ஸ் வந்து நடக்குது. யூகேல சீக்வன்சிங்ல காட்டியிருக்காங்க தட் ஈவன் அமங்ஸ் த ஹியூமன் சீக்வன்சஸ் சம் ரீகாம்பினேஷன் ஹேப்பன்ஸ் இதுல. ஸோ இது வந்து பேட்ஸ்ல தான் பெரும்பாலும் இப்போ இன்டர்மீடியேட்டுங்கிறது வந்து அவசியம் இல்லை அப்படின்னா சார்ஸுக்கும் ஃபீலிங். இப்போ சார்ஸ் பாம் சிவெட் அப்படின்னு சொல்லிட்டுனாங்க. ஆனா இப்போ பாக்குற இதுல வந்து even SARS and SARS related viruses and bats are very close. நான் அதை ஒரு ஸ்லைட்ல காட்டினேன், அதை தான் அது. அதனால இன்டர்மீடியட் பேட்ல இருந்தே நேரம் இப்போ எபோலா வைரஸ்லாம் பேட்ல இருந்து நேரம் வருதுன்னு தான் ஒரு இது. அதுலயும் வந்து இன்டர்மீடியட் பல வருஷங்களாவே நமக்கு தெரியாது இன்டர்மீடியட் இருக்கா, இன்டர்மீடியட் இது என்ன அப்படின்னு. அதே போல இந்த மார்பர்க் வைரஸ் 67 வந்தது கூட நமக்கு வந்து அது மங்கி வழியா வந்திருக்கலாம். பட் மீதி பேருக்கு இப்போ மார்பர்க் வைரஸ் நிறைய பேர் ஆப்பிரிக்காலே இன்ஃபெக்ட் ஆறாங்க. அவங்களுக்கு வந்து நேர பேட்ல இருந்து வருதான்னு பார்க்க தான் இப்போ வந்து பேட்ஸை டேக் பண்ணி அது போற இடமும் அது இன்ஃபெக்ஷன் வர இடமும் ஒன்னா மேட்ச் ஆகுதான்னு பாக்குறாங்க. பேட்ஸும் ஒரு அனிமல் தான். அதனால பேட்ஸ்ல இருந்து நமக்கு இப்போ நைப்பா வைரஸ் வந்து நேர பேட்ல இருந்து தான் நம்ம கேரளால வந்தது, இன்டர்மீடியட் ஒன்றும் கிடையாது. அப்போ இந்த SARS-CoV-2க்கு இன்டர்மீடியட் அனிமல் தேவையாங்கிற ஒரு கேள்வியே வந்து இப்ப நிறைய இருக்காங்க. ஏன்னா பேட்ஸ்லயே அது நிறைய பாப்புலேஷன் இருக்கு, சர்க்குலேஷன் நடக்குது, பேட்ஸ்லயே ரீகாம்பினேஷன் நடக்குது. எப்படி ஹியூமன்ஸ்ல நடக்கிற மாதிரி பேட்ஸ்லயே ரீகாம்பினேஷன் நடக்குது. அதனால அதுல இருந்து நேர வர வாய்ப்புகள் இருக்கலாம் அப்படின்னு தான் இப்ப பாக்குறாங்க. அதான் நான் சொன்னேன், சார்ஸும் அப்படிதான். மெர்ஸ் கூட கேமல் நாங்க, ஆனா இப்போ 2015 பண்ணதுல பாக்குறாங்க கேமல்ல இருந்து ஹியூமன்ஸுக்கு ஸ்ப்ரெட் பண்ண வாய்ப்பு ரொம்ப கம்மியா இருக்குன்னு, டிரான்ஸ்மிஷனே எஃபிகசியே இல்லைன்னு. எப்படி வந்து கேமல்ல இருந்து வந்திருக்கும் அப்போ அதனால அந்த கேள்விகள் மறுபடியும் வருகிறது. அப்போ அது வந்து நம்ம அதை நான் HIV 1க்கு சொன்ன மாதிரி 20 வருஷம் ஆச்சு அது ஒரு குரங்கு கிட்டே இருந்து வந்தது, எந்த குரங்கு ஸ்பீஷிஸ் மூலமா வந்ததுன்னு கண்டுபிடிக்கிறதுக்கு. அதுவும் ஒரு தற்செயலா ஒரு லக்கி பிரேக்னால கிடைச்சது. இப்போ அந்த மாதிரி நமக்கு வந்து பல வருடங்கள் ஆகலாம் அப்படின்னு தான் இப்போ கருத்து. அதனால இன்டர்மீடியட் ஹோஸ்ட் உடனே சுட சுட தோசை கிடைக்கலங்கிறதுனால அது வந்து இது என்ன ஆர்டிபிஷியலா பண்ண தோசைன்னு சொல்ல தேவையில்லை.
One question from Anthony King's Suresh Babu. Are the antibodies found because of actual COVID virus and by different vaccines same?
அத நான் அவரு ஜாம் டாக் முடிச்ச உடனே அதை பத்தி கேட்டிருந்தாரு, நான் அதை விளக்கினேன். அதாவது ஆன்டிபாடிஸ் வந்து நம்ம கோவேக்சின் போல இன்ஆக்டிவேட்டட் வைரஸ் அதுல இருந்து ஃபார்ம் பண்ற ஆன்டிபாடிஸ் வந்து வெரி சிமிலர் டு ரேஞ்ச் ஆஃப் ஆன்டிபாடிஸ் ஃபார்ம்ட் அகேன்ஸ்ட் டிஃபரெண்ட் ப்ரோட்டீன்ஸ் ஆஃப் த கொரோனா வைரஸ். ஆனா இந்த ஸ்பைக் புரோட்டீனை வச்சு பண்ற வேக்சின், கோவிஷீல்டு, mRNA வேக்சின், ஸ்புட்னிக், இது எல்லாமே வந்து ஆன்டிபாடிஸ் எகெயின்ஸ்ட் ஸ்பைக் புரோட்டீன் தான் பண்ண முடியும். ஏன்னா அதுதான் நம்ம ப்ரெசென்ட் பண்றோம். அது ஸ்பைக் புரோட்டீன் இம்பார்ட்டண்டா இருக்கிறதுனால அதுவே வந்து பெரும்பாலும் அது எஃபெக்டிவா இருக்குன்னு தான் பாக்குறாங்க.
There are two people who have raised hands. Aditya.
Yeah.
Udayan and Aditya.
அன்மியூட் பண்ணனும். ரெண்டு பேருமே மியூட்ல இருக்காங்க.
Yeah, can you hear me?
Yeah.
Yeah, like first of all, like I like this talk very much, and the congratulations also, because it's very much necessary at this point of time. I have just two comments. Like, first of all, we should understand this in the larger perspective. Like at present in our country, there is some anti-China sentiments going. From that perspective only, we should understand this also, because if you see, like, for the past 1 year, since we are just serving for the American imperialists, like this anti-China sentiments is going everywhere in the national media. So only this topic has become a like heavy debate everywhere. That's the first thing. Second thing is, like, as you mentioned about the zoonogenesis, like I want to understand even more. Like, if you see in all the viruses which have been evolved, like in the past 1 or 2 decades, either from the 20th, like 1997, there was H5N1 virus, and after that there was HN11 and SARS, everything. Like, you see, there was the intermediate host is either being a pig or some like chicken or like many other animals. So, like, how should we understand this? Because I see in this perspective, like this poultry farming and other things has been majorly developed over the years. Like, they have been increased in vast percentage over the years, and that is why, like, since we are being and destroying this forest and other things in the human living regions, so these animals comes to the human living region for their food and other things. So that's why, if you see in the history for the past few years, like every virus has been transmitted through this zoonogenesis only. So actually, by speaking about the science also, we have to speak in this perspective, like how to control this vast development of this poultry and other things. Then only we can limit ourselves in future to there won't be many other viruses coming. See, if you, before the COVID virus gets down, there is Zika virus, other things going on. So in future too, there will be many other such viruses. So I think you can just put in this perspective also. I love to hear your answer.
ஆதித்யா, நீங்க ஃபர்ஸ்ட் ஸ்லைட்ல பாத்தீங்கன்னா, நான் ஃபர்ஸ்ட் 4 ஸ்லைட்ல வந்து அதை தான் சொல்லியிருந்தேன். உங்களுக்கு வந்து எப்படி ஹியூமன் பீயிங்ஸ் நிறைய ஆக்குபை பண்றோம், டொமஸ்டிக் அனிமல்ஸ் நிறைய இருக்கிறதுனால ஆட்டோமேட்டிக்கா ஸ்ப்ரெட் பண்றது கூட இருக்கும். அண்ட் ஹெவிலி பாப்புலேட்டட் ஏரியாஸ் லைக் சைனா அண்ட் இந்தியா வந்து மோர் ஜூனோட்டிக் ஆரிஜின்ஸ் வர வாய்ப்பு இருக்குன்னு. அந்த ஈக்கலாஜிக்கல் இம்பேலன்ஸ் வரும்போது வர வாய்ப்புகள் கூடுதலா இருக்கு. அதனால நம்ம வந்து அத என்ன all development requires some amount of இது. அதனால you cannot say டெவலப்மெண்டே கூடாது அப்படின்னு இல்லை. அது வந்து நம்ம பேலன்ஸ் பண்ணணும். அதான் மெயினா இருக்கு.
அனைவருக்கும் மதிய நேர செந்தமிழ் அறிவியல் வணக்கம். நான் பேச கேக்குதுங்களா?
கேக்குது.
இந்த உரை விஞ்ஞானிகள் முனைவர் T.R.Govindarajan, முனைவர் எஸ். கிருஷ்ணசாமி ஒரு உரை ரொம்ப அற்புதமா இருந்துச்சு. எனக்கு ஒரு 2 கேள்விகள். ஒன்று, கோவாக்சின் வந்து அந்த 3rd ட்ரையல் பண்ணுறதுக்கு முன்னாலேயே வந்து அதை நம்ம பயன்படுத்திட்டு இருக்கோம். இன்னும் கூட WHO வந்து அதை முறைப்படி முழுமையாக அங்கீகரிக்கலை. இப்போது கோவாக்சின் போட்டால் நீங்கள் வந்து அமெரிக்கா போன ஐரோப்பிய நாடுகளில் வந்து அனுமதி இல்லை அப்படிங்கிற நிலைமை இருக்குது. இப்போது ஏதோ ஒரு காரணத்தினால 3rd ட்ரையலில் வந்து இது வேண்டான்னு ஒரு முடிவு எடுத்திருந்தா நம்ம நிலைமை என்ன ஆகும்? நம்ம ஏன் இப்படி அவசரப்பட்டுருக்கலாமா? தப்பி தவிர இப்போ வந்திருந்தால் என்ன ஆயிருந்திருக்கும்? நான் வந்து ஒரு ஹைப்போதெட்டிக்கல் கொஸ்டின் தான், இப்படி ஆயிருந்தால். பட் அதையும் நம்ம வந்து ஒரு ப்ராபபிலிட்டி நிகழ்தகவுல நம்ம பார்க்கணுமா இல்லையா? ஒரு கேள்வி. 2வது வந்து நம்ம வந்து உருமாறுது, வேரியன்ட்ஸ் வருது. வேரியன்ட்ஸ்க்கும் வந்து டக்குன்னு சொல்லிடுறாங்க, இந்த கோவாக்சின் நல்லா இருக்கு, கோவிஷீல்டு நல்லா இருக்கு, பைசர் நல்லா இருக்கு, டக்குன்னு சொல்றாங்க. உருமாறின பிறகு ஒரு நீண்ட ட்ரையல்ஸ் பார்க்கணுங்கிறது தான் நான் ஒரு லேமேனாக, சாதாரண மனிதராக நான் பார்க்குறது. மிக விரைவாக சொல்லிடுறாங்க. இதில் வந்து ஏதோ கார்பரேட் நலன்கள், ட்ரக் இண்டஸ்ட்ரியுடைய மாஃபியாவுடைய நலன்கள் இருக்கிற மாதிரி ஒரு ஃபீலிங் இருக்கு. இதெல்லாம் வந்து சயின்டிஃபிக் கம்யூனிட்டி கொஞ்சம் டைம் எடுத்துட்டு சொல்லணும்னு நான் நினைக்கிறேன். அவ்வளோ விரைவாக வந்து சொல்ல முடியும்னு எனக்கு தோணலை. ஏன்னா பல வேக்சின்ஸ் வந்து 6 வருஷம் 10 வருஷம் தேவைப்படுது. பட் முன்னர் கிருஷ்ணசாமி வந்து பேரலாக நிறைய நடந்ததுனால சீக்கிரமாக வேக்சின்ஸ் கண்டுபிடிச்சேன்னு சொன்னாலும் கூட வேரியன்ட்ஸ் வருது. அந்த வேரியன்ட்ஸ்க்கு வந்து அவ்வளோ சீக்கிரமாக வந்து இதுக்கும் நாங்கள் வேக்சின் அப்ளை ஆகும் வந்து
இதுல மறுபடியும் நீங்க வந்து வேரியன்ஸ் வருதுனாலும் நீங்க அந்த வேரியன்ஸை வந்து பிகாஸ் நம்ம பேப்பர்ல ரிப்போர்ட் ஆகிறதுக்கும் சீக்வன்ஸ் டேட்டா பேஸ்ல ரிப்போர்ட் ஆகிறதுக்கும் முன்னாடியே இருக்கிறனால அதை பீப்புள் முன்னாடியே டெஸ்ட் பண்ண ஆரம்பிக்கிறாங்க. நிறைய பேர் இதுல வந்து இப்போ முக்கியமா இந்த டயத்துல பாத்தீங்கன்னா லாட் ஆஃப் ஓப்பன் ஷேரிங் ஆஃப் டேட்டா இருக்கிறதுனால டெஸ்டிங் வந்து மிக வேகமா நடக்குது. இது வந்து உடனே வந்து நம்ம மாஃபியா, கார்பரேட் மாஃபியா அப்படின்னு போக அவசியம் இல்லை. இதுல வந்து நம்ம கவர்மெண்ட் வந்து சில கவர்மெண்ட் கரெக்டா பண்றாங்க, சில கவர்மெண்ட் நம்ம கவர்மெண்ட் வந்து ரொம்ப ப்ராப்பர் ப்ரிகாஷனோட எல்லாம் பண்றாங்க. பட் ப்ராப்பர் ப்ரிகாஷனோட பண்ணுமே தவிர டெஸ்ட் பண்ண பிறகுதான் டேட்டா அனௌன்ஸ் பண்றாங்க. அதனால மேபி நம்ம பிரஸ் மீட்ல சில டேட்டா அனௌன்ஸ் பண்றாங்க, அதுல வந்து அர்த்தம் இல்லாம இருக்கலாம். பட் நம்ம கேட்க வேண்டியது என்னன்னா, எந்த டேட்டாவா இருந்தாலும் பப்ளிக்ல அந்த டேட்டா ஷுட் பி மேட் அவைலபிள், டெஸ்டிங் டேட்டா ஷுட் பி மேட் அவைலபிள். அதான் நம்ம இதுன்னு நீங்க அதை வந்து கார்பரேட் மாஃபியான்னு வந்து லேபிள் பண்ண அவசியம் இல்லைங்கிறது கருத்து.
I just wanted to add, முக்கியமா அதுல வந்து ஈவன் கோவேக்சின்க்கு எல்லா இதுலயுமே எமர்ஜென்சி யூஸ் தான். Unlike the previous vaccines where it is tested over a long time, etc. That's why it takes 2 or 3 years, 5 years, etc. Here we have to produce something, we have to protect. So there is some emergency use authorization which is happening in all the countries. All the vaccines have been given only emergency use vaccine. Now Covaxin, there is some initial problem about the third phase not completed. And because other things have come and there's a number of volunteers, etc., there was some problem, etc. But as of now, the data is available even for Covaxin. The government has given out, and it is also getting peer reviewed. And I do not anticipate a serious problem. It may have, in comparison with Covishield or Pfizer or other things, may have less efficacy, as they call it. Okay, it may have or may not, and we may not even know whether it is better even for the variants which are happening, etc. Now, this is the situation as of now, and we need not go about suspecting either the government authorities or the scientists that they are bypassing for corporate purposes. We don't have to do that kind of a thing. We have to ask, be transparent, give the data to be analyzed by all the scientists, not from me. I'm not a scientist who are working on this thing, by the appropriate people who are working on this, so that the data is publicly available. They can scrutinize and get better understanding of that. That is what I would like to say.
என்னுடைய இதுன்னா இப்போ அவங்க வந்து இப்போ ஒரு இந்த கவர்மெண்ட் வந்து டிஆர்டிஓடைய அந்த D2 குளுக்கோஸ் அப்படிங்கிறத வந்து சமூக வலைதள சோசியல் மீடியா என்ன சொன்னாங்கன்னா, இந்தியா வந்து ஒரு மிகப்பெரிய அளவில் கண்டுபிடிச்சிச்சு, அந்த COVID-19 வைரஸே வந்து ப்ராப்ளமே சால்வ் ஆகிடும், உலகத்துக்கே ஒரு பெரிய நன்கொடை, உலகத்துக்கே சரியா போயிடும் அப்படின்னு சொல்லிட்டு பயங்கரமாக வந்து பில்டப் கொடுத்து பண்ணாங்க. பட் நான் விக்கிபீடியாவில் பார்த்தபோது இட் இஸ் அன் அட்ஜங்க்ட், இது ஒரு துணை மருந்து, அதுவும் வந்து ஒரு மருத்துவருடைய அவசர காலத்துக்கு தான் பயன்படுத்தணும், மருத்துவருடைய ஆலோசனை தான் பயன்படுத்தணும்னு சொல்லுவேன். இன்னும் கூட அது பிரபலமாக மக்கள் மத்தியில் போகலை. அது 990 வேலை சொல்லியிருக்காங்க. இந்த மாதிரியான இப்போ டிஆர்டிஓ வந்து அந்த பண்ணக்கூடிய விஷயங்களை வந்து அவங்க வந்து இது அட்ஜங்க்ட், இது வந்து அப்படின்னு ஒரு அவங்களே வந்து இப்படிலாம் வந்து மீடியாவில் வர்றது வந்து சரியில்லை அப்படிங்கிற மாதிரி ஒரு அரசாங்கம் வந்து தவறான பிரச்சாரம் பண்ணப்படுது, இப்படிப்பட்ட பிரச்சாரங்கள் பண்ணக்கூடாதுன்னு சொல்லிட்டு அரசாங்க தரப்புல இருந்து ஒரு மறுப்பு கூட வரல. இந்த அரசாங்கம் வந்து நம்ம எதிர்பார்த்த அளவுக்கு இல்லைங்கிறதையும் நம்ம பார்க்கும்போது தான் வந்து கோவாக்சின் வந்து தேர்ட் ட்ரையல்ஸ் பண்ணாத போது நமக்கு பயம் வருதுன்னு நான் நினைக்கிறேன்.
இல்ல உதயன், கோவேக்சின் தேர்ட் இது ஃபேஸ் ட்ரையல் பண்ணிட்டாங்க. அது இன்னும் டேட்டா பப்ளிஷ் பண்ணல, அவ்வளவுதான். அதுல வந்து அதுக்கு இப்போ நான் பப்ளிஷ் பண்ணணுங்கிறது அவசியம். அத இதுல 2DGல நிறைய குழப்பம் பண்ணாங்க, தேவையில்லாம ப்ரொபகண்டா பண்ணாங்க. ஏன்னா அப்போ வந்து அவங்களுக்கு செகண்ட் வேவ் நிறைய பாதிப்பு இருக்குனால டைவர்ட் பண்றதுக்காக பண்ணாங்க. நம்ம என்ன பிரிச்சு பார்க்கணும்னா, அரசாங்கம் பண்ற கோமாளித்தனத்துக்கும் நம்ம உண்மையான ட்ரையல்ஸ் பண்ணி பண்றதுக்கும் பிரிச்சு பார்க்கணும். நம்ம கோரிக்கை என்ன இருக்கணும்னா, எல்லா இதுவா இருந்தாலும் ட்ரையல்ஸ் பப்ளிக்கா டேட்டா அவைலபிளா இருக்கணும், அது எல்லாருக்கும் ஓப்பன்னஸ் இருக்கணும். அதான் இதுலன்னு அதை இந்த மாதிரி அரசாங்கம் அப்போஸ் பண்றதுக்கு மீதி பேர் வந்து அப்போஸ் பண்ணி பேசணும் இல்லையா? சோ அது ஏன் இப்போ வந்து அரசு கோமாளித்தனத்தை வந்து எந்த பெரிய நிறுவனங்களோ அல்லது இவங்களும் அப்போஸ் பண்ணல, மீடியாவும் இது பண்ணல. ஏன்னா அவங்களுக்கு அதுல இது இருக்கு. அதனால நீங்க அரசோட இதுக்கும் இது வந்து நம்ம அதனால மக்களை வந்து தவறான பாதையில போக முடியாது இல்லையா? நம்ம அரசு கோமாளித்தனமா இருக்கு, துக்ளக் தர்பாரா இருக்கு, அதுக்காக நம்ம வந்து இதை கொண்டு வர முடியாது.
ஒரே ஒரு ஸ்லைட் கரெக்ஷன். The preprint on Covaxin is available. Now it has not been peer reviewed. It has not been peer reviewed. Secondly, they have also submitted to WHO, World Health Organization. They have asked for clarifications. Now these are all processes which can take some time. Okay. Even Covishield, which is recognized by WHO, now European Union is not yet recognized it. So, for example, using Covishield, you cannot go to Europe that I have been vaccinated. You can go to UK, but not to Europe, the Germany and other things, because of this thing which is there. So there are complications of this kind will be always there. So there is some international collaboration and cooperation which is happening now on this, which is far better than many other things. We have to understand that. So that is what is the crucial issue which we have to appreciate. Next question, Nadarka.
Sir, can you hear me?
Yes, Arun.
Yeah, sorry, I've been chatting all the while. I had my other work also. First of all, kudos to you all, sir. You're doing a fantastic job, doing a very good presentation. I've been following you all for the last couple of years. It's really good. But then, sir, I beg to differ. You know, WHO is an organization which is funded by money, and the pharma is an industry, and industry will function only if they make profits, not from human lives. Okay, because I'm also in the industry, and I do have friends in the pharma industry. So I'm just being very open and very sharing with you openly. So if an industry has to survive, they need profits. Come hell or high water, birth or death rates, they need profits. So I doubt if the farm industry is going to do something to, save lives. They will do things only with which they can sell more. The more slow the vaccine process comes out, the more the money they can make, the more vaccines they can sell. I mean, it's they may be first dose, second dose, third dose. The doses will just keep on going so they can sell more, plain and simple. So, of course, this is my take, and I could be wrong though, but I'm just sharing my 2 cents with you, sir.
Okay. Just there is a minor, there is a difference. WHO is under United Nations. World Health Organization comes under that. And there is, as you mentioned, there is some international corporates and thing, etc. through their governments play some role. There is no doubt. They do that even in United Nations. They do that in Security Council. That is part of our life systems. But there are people outside this thing who talk about and debate and come to some conclusions and expose the pharmaceutical companies also. And if they get exposed, it may not be advantageous even to make money if it is too much exposed. So they will also have to be careful about it. So there are some issues of that very complicated political and scientific nature which is going on in this country, which is beyond many of us. Let me make it clear. We are only a small fry in all this small thing in this whole game. So given all that, as people, what is it we can ask and we can expect and we can get some results out of it is the clarity which is required. And at the moment, the World Health Organization is presenting some details involving discussions between scientists from various places as represented by their countries, including India and other countries. So we have to go by that, and then we can even question World Health Organization. You don't have to agree with them. We don't have to agree with them. We don't have to agree with scientists also. Science is something which will be very happy to accept disagreement with the science itself. There is no, we are not religion. It's not religion, right? So that is the point which you have to appreciate.
True, so agreed. What he says is right. I agree with you.
I think
almost 1 o'clock.
Yeah, I think we can, at some stage we can close.
வேற ஏதாவது கருத்துக்கள் இருந்தா சொல்லணுமா?
இல்ல க்ளோஸ் பண்ணிடலாம், அவ்வளவுதான். எல்லா ஆல்மோஸ்ட் ஆல் क्वेश्चंस ஆர் ஆன்சர்ட்.
லாஸ்ட் கால். ஓகே, அனைவருக்கும் நன்றி. நல்ல பல விளக்கங்களையும் விவாதத்தை தொடங்கி வச்ச பேராசிரியர் கிருஷ்ணசாமி அவர்களுக்கும் கோவிந்தராஜன் அவர்களுக்கும் நம்முடைய மனமார்ந்த நன்றி. அதான் முதல்ல சொன்னது மீண்டும் வலியுறுத்தி விரும்புகிறேன். இது என்ன, இது ஒரு விவாத மேடை. அந்த விவாதம் ரொம்ப முக்கியம். அறிவியலுக்கும் சமூகத்துக்கும் இருக்கக்கூடிய தொடர்பு என்பது இந்த மாதிரியான வெளிப்பாடுகளில் நிறைய பாதிக்குதுங்கிறது நமக்கு தெரியுது. இந்த கொரோனா பெருந்தொற்று, அதை நம்ம சந்திக்கிற விதம், இது எல்லாவற்றிலையும் அறிவியல் கண்ணோட்டம், அறிவியல் விழிப்புணர்வு மிக மிக அவசியம். அந்த அடிப்படையில் தான் இந்த விவாதங்கள் நடைபெறுகின்றன. தமிழ்நாடு சயின்ஸ் ஃபோரம் வெல்கம்ஸ் everyone to take part in these discussions and contribute to the discussions. I think we need a lot more evidence-based discussion in our society. So I thank you all for participating in this discussion.
Okay,
thank you.
Thanks, Jam. Thanks, Vijayan. Thanks, everybody.
Yeah, thanks, Vijayan.
Okay.